First of all, I want to thank you, Jeff, and all of the organizing committee for today, as well as the guests for coming. Very interesting talk. Thank you very much. So I'm going to talk to you about the first part of my work during my PhD, which was to really troubleshoot the map, the magnified analysis of the proteome that Kaylee used, and also Bettina is using in her workflow. And so the experience with this technique is this one, and also expansion microscopy, is even though the principle is quite simple, it's quite hard and requires a lot of troubleshooting to put it in place reliably and reproducibly, and especially with the initial protocols as well as paper that has been published, because it was not very detailed and not very adapted for not bioengineering. So I spent quite a bit of time to try to adapt this procedure for the study of excitatory synaptic proteins. So first of all, why study synapses? So they are the central structure or a central structure of the brain of intercellular communications between neurons, and they are extremely complex in their protein content as well as in their density. Just a warning, what I'm going to show you, I don't even understanding myself, but like they are packed with multiple proteins, thousands of proteins, all interacting together in order to process the information from one side of the presynaptic side to the postsynaptic side. And this requires thousands and thousands of proteins. And all of this is within, yes, so all of this is within 100 nanometers between the presynaptic compartment and the postsynaptic compartment, which makes it very difficult to access each and every single of these proteins, especially in tissue and in thick tissue. The gold standard has long been electron microscopy, where you can see the postsynaptic density here, very dark, but the limitation of electron microscopy is we cannot really target specific proteins, or you can do it with gold particle, but only one protein at a time. You cannot really do multiplexing. And also the amount of volume that you can image, acquire, and analyze is quite small if you want to spend a reasonable amount of time and not spending like years of doing this processing. Light microscopy is faster, allows to image bigger volume faster. However, it has other limitations. It's limited in its resolution, especially standard confocal microscope. But even if when you have access to strong super resolution microscopes, instead, if you cannot access the antigen of interest and their synaptic proteins, you cannot really decipher the different proteins within this subcellular compartment. So we have been working on the magnified analysis of the proteome to establish a reliable and easy to use protocol for people to use and especially study excitatory synaptic proteins. I'm not going to go over in too much detail about the protocol. This has been published and it's available to everyone. So to look at the step-by-step protocol, I would refer you to this presenting the paper that I'm going to show you. Just to go quickly over the procedure, because they have been talking about this previously. The principle is to make the tissue physically bigger by embedding it in a polymer of acrylamide, and then by some denaturator and washing, you get rid of most of the lipids, and so which makes it more accessible for the antibodies to target the antigens of interests. So we gathered all of these troubleshooting in this paper that has been recently published. And as I was mentioning, there is a protocol for employing expansion microscopy, the magnified analysis of the proteome, very detailed step by step, and also how to stock the different stock solution. So I hope this could help people to implement this procedure if needed or if they want. Once it's troubleshoot after a couple of weeks or months, you can start getting reliable results out of this protocol. So this paper, there was three main goals. First, to characterize the map performances across different steps, as well as different microscopes. Then to test also map in different conditions, because not all of our tissue, we want to use it only with expansion microscopy. We may want to use it for other experiments, or we may also get our tissue from other experiments. So to see if we could combine the map procedure with different conditions of fixation. And then assess the ability of MAP to spatially profile synaptic proteome quantitatively, because at the end, we want to generate quantitative data to investigate biological questions. And so the result I'm going to show you has been acquired in the CA1 subregion of the hippocampus, and especially in the stratum radiatum, which is very dense in excitatory synapses. So to show how powerful is this technique to locate proteins in a very densely connected region. So at first, for the first two goals, we used two different antibody combinations. For the sake of time, I'm only going to show you the results for one, which targeted the voltage-gated calcium channel CAV 2.1, the presynaptic active zone component ELX, and an antibody targeting all of the MAGUC protein in the postsynaptic density. I'm choosing this antibody combination because they have a it's very nice to show how with this procedure we can decipher the presynaptic compartment from the postsynaptic compartment. And at the best, we can even look at what's happening between this pre and postsynaptic compartment, looking at the calcium channel. So first, about the characterization of MAP, I'm going to go quickly on this. We're going to show what is the gain of synaptic protein visualization. And as Katie showed you before, we have a real improvement in this visualization. With regular IHC, looking at these proteins, with thick tissue sections, it's quite hard to see if what we are seeing is specific or if it's just a lot of background, especially when we look at the MAGUC proteins, which are a marker of excitatory synaptic proteins. We should see them only in the stratum radiatum subregion, while we can see here that the staining, the labeling goes also over the pyramidal layer that has more inhibitory input and less excitatory input. Once we process the brain with the MAP procedure, this time we can see a sharp difference, and especially a sharp difference in the labeling between the stratum radiatum and the pyramidale. We can see this excitatory input is mainly present in the stratum radiatum as expected. And if we get a closer look, and we want to look at the individual synapse level, we can see that only MAP processed tissue for this thick tissue section allows to resolve the synaptic protein at the individual synapse level. Looking at the quantification, we wanted to see if this was not just a random colocalization of this protein. So what we did is we look at the ratio of clusters that are colocalizing with one another for all of the proteins showed here. And we can see that when we look at MAP processed tissue, most of the proteins are colocalizing with one another, compared to regular IHC procedure, where we have a very low amount of the cluster that I could label individually that are not colocalizing at high rates. And to see if this colocalization was just random or not, I just flipped one channel compared to the other channel to see if we still have this high amount of colocalization. And we can see that once we do this for the MAP processed tissue, we reduce drastically the amount of protein that are colocalizing with one another. While when we do this with standardly processed with IHC section, they present as much colocalization as when not flipped. So showing the power of MAP to allow to have this individual synapse level labeling of synaptic proteins. Then we wanted to see at which step of the procedure we get this improvement in synaptic visualization, because there are two main steps that could be important. One would be the antigen accessibility. In the MAP procedure that we are employing, we first do the dilation, then the delipidation, and only after the delipidation, we do the antigen labeling with antibodies. So by doing this delipidation, we could have a gain of antigen accessibility that would allow to obtain this improvement in synaptic protein visualization, which we can see here. So in this row, a tissue has been map processed, but not expanded and labeled for the proteins of interest. And so we just incubate this tissue in regular PBS. So keep it, so it's a bit expanded, 1.4, 1.5x, but it's not fully expanded 4 times. And we can still see that at that level of expansion, we can see individual clusters of penmagogue that are aligning in front of us, and in some having calcium channel lying between the pre and the post-synaptic marker. Then we looked at the gain of visualization once we expanded physically this tissue. And so what we observe first is if we use exactly the same imaging settings to look at an expanded tissue, the exact same tissue but expanded, we cannot see anything due to the dilution of the signal. So it shows here that by expanding physically the tissue, we pull further apart the different fluorophore, which dilute a lot the signal. So it requires a higher laser intensity and a higher gain to be able to pinpoint this individual signal, individual cluster. But once it's optimized, we can also see individual clusters, but we get a bit more detail into the distribution of these synaptic clusters. And especially here, we can see that for the green signal, ULX, there are multiple clusters that are aligning in front of one penmagogue signal. And this could be related to these nano clusters that has been shown recently. Synaptic proteins are not homogeneously distributed. They are more clustered in these kind of tiny nano clusters, and we can start to touch them, see them using just expansion microscopy without having to use any fancy super resolution microscope. Then talking about super resolution microscopy, we wanted to see if by combining MAP with various microscope, and from regular confocal microscope to super resolution microscope, we could get a bit more information about this distribution at a nanoscale. And so what we can see looking here at PSD95, which is another postsynaptic density protein, when we just image a map processed tissue with a regular confocal microscope, we can still see them at the individual synapse cluster. But once we use a type of super resolution microscope, it's not actually a super resolution microscope, but it's more on the processing aspect. We can start to decipher a bit more the nano-organization within the synaptic cluster on the postsynaptic side. And then this is another type of super resolution microscope, which has a bit more processing, a bit more detector, and we can see the nanocluster even more. And then when we use a true super resolution microscope, instead, we can definitely see all of these nanocluster on the postsynaptic side way more than with a regular confocal microscope. Here, compared to the images before that I showed you, we are looking at the postsynaptic side, which is a bit more difficult because more densely packed to see all of the nanoclusters. And we quantified the resolution that can be achieved using these different microscope and the expansion microscopy. And what we can see here is using regular confocal microscope, we can achieve a resolution between 60 to 100 nanometer of resolution, while with the Eriscan, we can almost achieve 25 nanometer of resolution, while instead using the magnified analysis of the proteome, we can go all the way to 15 nanometer of resolution. So then once characterized, we wanted to see if we could apply this technique in different, with tissue fixed in different initial fixation condition. in the original map paper, they were using this kind of fixative that was containing acrylamide and PFA in the same solution, but this solution is quite tedious to perfuse directly in the animal because it's very viscous. So, and also once you use this kind of fixative, you cannot use your brain for other experiments. So then we've tried to fix the animal with regular PFA fixation. So then the brain could be used to perform any other type of experiment that you would like. We also tried to directly extract the brain and incubating it in this map fixative solution, which could reproduce condition in which people would do IFIs and then would just want to put their sample in the fixative solution and see if they could use the expansion microscopy to perform to do more in-depth analysis. And then finally, the last condition, we're using formalin, which is the fixative that is commonly used to fix human brain section for like 1 week to 1 month, because human brain tissue are preserved in this fixative for an extended period of time before being embedded into paraffin. And we tried to mimic also the condition, the post-mortem condition, where the brain is not directly fixed, so it's extracted and stayed at 4 degrees for a couple of hours before being fixed in the map fixative solution. And I think one thing that we can already see is like qualitatively, all of the synaptic ponta can be resolved at the individual synapse level. Then looking at the quantification of the signal to background ratio, for all of the condition, the signal to background ratio is quite high, on the order of, on average, 10. Even though looking at the two-way ANOVA, there is a significant difference in the fixation tab as well as in the antigen. And then looking at the variation of the signal to see if the signal varies more in one condition than another, it is quite homogeneous, or at least quite at the same level for all of the conditions, even though there are still significant difference between the different conditions and the different antigen. Then we also wanted to try to use this on FFPE mouse brain section. So FFPE means formalin fixed, then paraffin embedded mouse brain tissue. These are the exact conditions that are used for banking human brain after either resected surgery or postmortem surgery. And so we wanted to see if we could apply MAP on tissue preserved in such condition, which would open a panel of possibility of using the biobank immense. So what we can see here is like at the large scale level, we have the right pattern of distribution of these different synaptic markers. And looking at the individual synapse level, we can see that all of the synapse proteins can be locked at the individual synapse level. So finally, we wanted to see if this technique could be quantitatively used to map the profile, the synaptic proteome profile. And so for this, we decided to look at the difference of excitatory input made on interneuron and pyramidal neurons in the hippocampus, the same region that I was talking to you before. And so for this, we used a transgenic mouse that is expressing YFP, the channel rhodopsin tagged to YFP in all hippocampal interneurons. So using this marker, we could look at synapses made on these dendrites or out of these dendrites, that would be the pyramidal neurons. And then we could look at PunMagook to locate all of the excitatory inputs in this field of view. And then looking at AMPA receptors as well as glutamatergic receptors and see if they have a differential distribution. This has been already showed before using electron microscopy, but we wanted to see if we could reproduce the same result that has been acquired before with electron microscopy using this procedure. So we can see here already that for AMPA receptors, you have brighter clusters, especially at bigger synapses compared to the rest of the section. While for glutamatergic, the NMDA receptors, it's more homogeneous and maybe a bit fainter for the synapses made directly on the dendrites. So when we looked at the quantification, first, when we looked at the size of the excitatory synapse made onto these dendrites, We could see that dendrites, synapses made on interneurons are a bit bigger than synapses made on pyramidal neurons, looking at the volume of the pan-MAGOC. While looking at the mean intensity of the fluorescent signal for pan-MAGOC, there are no differences between synapses made on interneuron or pyramidal neurons, which is expected because the density of pan-MAGOC should not be different on bigger or smaller synapses. Then looking at AMPA receptors, we observed quantitatively the difference that we were seeing qualitatively with a higher sum intensity, so total amount of AMPA receptors, at synapses made on interneurons, as well as mean intensity, so density of AMPA receptors in synapses made on interneuron. While the opposite trend was observed for glutamatergic receptors, especially for the mean intensity. The mean intensity looked way higher on synapses made on pyramidal neuron than synapses made on interneuron. So again, all of these results were known before using EM data, but this time we use the MAP procedure, and which is way less tedious to put in place and way faster to acquire and analyze data than electron microscopy. And then we wanted to go beyond the glutamatergic receptor because we know now that the postsynaptic content, especially at the postsynaptic density, is very heterogeneous in excitatory synapses. And so we wanted to have our focus on the Schenck family proteins because this family of proteins is known to be involved in a number of psychiatric disorders, including schizophrenia and autism. But yet we didn't know if we didn't know yet if there was any differences in the input on different cell type. And using MAP, we investigated this. So using the same mouse as before, we looked at synapses made on interneurons versus synapses made on pyramidal neurons. And what we could find is only SHANK2 looked not to be expressed in synapses made on interneurons. And when we looked at the quantification, we could see that there was a significant difference in the mean intensity as well as the sum intensity for the Schenck signal on synapses made on pyramidal neuron versus synapses made on interneurons, while there was no difference in the mean intensity for synapses made in synapses containing Schenck1 and Schenck3. While for the sum intensity, the total intensity, it's significantly higher in synapses made on interneuron than pyramidal neuron, but this could potentially be explained by the fact that synapses in interneurons are bigger than synapses on pyramidal neurons with the data that I showed earlier. So here we show the ability of MAP to quantitatively map the profile of the synaptic content on synapses made on different cell type. And so to conclude, we could see that there is an improvement in antigen visualization, which is primarily due to a gain in antigen accessibility, and the physical magnification brings just more detail into the visualization of the distribution within synapses. MAP is also we're also able to use MAP in different conditions of fixation. And right now we are using MAP on PFA fixed mouse brain tissue as well as human brain tissue. And finally, we are able to spatially profile the synaptic proteome quantitatively using this technique. And so for the next step of my PhD, I would like now to use this procedure to investigate the synaptic proteome of human hippocampus, and especially its alteration in the context of epilepsy, here using, again, the MAP procedure to decipher all of the connectivity and all of the distribution of the synaptic proteins. So on this, I would like to thank my two labs, the Cembrowski lab as well as the Craig lab, for their support. And a big thanks to Jeff, but also Bank, who helped me a lot troubleshooting the MAP procedure and troubleshooting the whole pipeline to analyze this data. Thank you very much.